Hi, welcome to Biology classes with Lakshmi Chandra. Having completed the meaning and definition of biotechnology, let's focus on tools of biotechnology today. So these are the three important tools for biotechnology, especially for recombinant DNA technology or for genetic engineering, we call it as. So those three tools are, first one, restriction enzymes, cloning vectors, and then competent host. Once again, I'll say restriction enzymes, cloning vectors, and competent host. In today's session, we'll focus on the first tool of recombinant DNA technology, that is named as the restriction enzymes. Let's begin with this term, enzymes. Enzymes literally means in yeast. It literally means in yeast. What is it doing? It is also called as a biological catalyst. Now we all know catalysts, they enhance the rate of a reaction. They enhance the speed of a reaction. Now what is that? Let me just make it clear to you with a small example. Just imagine you are having 2 glasses of water, having equal quantities of water, and in both these glasses of water you are adding say 10 grams salt. In glass A as well as in glass B, you are adding 10 grams salt each. Now in the glass A, you are leaving it undisturbed likewise without any disturbance. In glass B, you are putting a spoon inside it and you are trying to stir it. Which one will dissolve? In which one the salt will dissolve faster? Of course, in the second one, in glass B, because you have added a spoon there and you are dissolving it. And when you take the spoon out, the shape or the size is nothing of the spoon changes, it remains as such, isn't it? But what the spoon has done? It has increased the speed of the reaction. That is, dissolution of salt in water has increased when you had stirred it with a spoon, but in the other one, which is kept undisturbed, still the process of dissolution is going on in a slower rate. The same thing is enzyme doing inside biological organisms, or that is the catalyst. It enhances all the reactions that is happening inside our body. But nothing happens to enzyme. After the reaction is over, the enzyme comes out unaffected and it is ready for the second cycle of reaction to happen. But enzymes are very, very, very specific. So, having understood the meaning of the term enzymes, let us see now what is this restriction enzymes. Restriction enzymes. Okay, let's see that. Restriction enzymes, they are called as molecular scissors. Molecular scissors. They are scissors, like they are used to cut molecules. They are molecules. Basically all enzymes are proteins, we know about that. And they are used to cut molecules. Isn't it interesting? Molecular scissors, restriction enzymes. But they cut only at specific sites. That is why restriction enzymes. So restriction enzymes, they cut molecules at specific sites. And these sites, these specific sites, where they go and cut, they are called as recognition sequences, or they are also called as restriction sites. Recognition sequences and restriction sites. So they are molecular scissors. Okay, they go and cut molecules at specific site. Now the question is, which molecules do they go and cut? For that, I have to name this restriction enzyme a bit more specifically. Let me name it. They are restriction endonuclease and restriction exonuclease. Restriction endonuclease and restriction exonuclease. So here you are coming across the term nuclease. Nuclease. Nuclease means enzymes which cut nucleic acid molecules. That is our DNA or RNA. Here in genetic engineering, we are focusing on DNA. Okay, so I told you restriction enzymes are molecular scissors which cut molecules at specific sites. Now we know those molecules are nucleic acid molecules. And the name is, I have modified it as restriction endonuclease, means they cut nucleic acids within. They make the cut within the nucleic acid molecules. This is restriction exonuclease, means they cut nucleic acids from their ends. In a very simple manner, I'll just tell you. For example, imagine we are cutting vegetables. Okay, if you start cutting from one end, it is equivalent to exonuclease. Cutting is happening from one end to the other end. But if we start cutting at the center or the middle of the vegetable, just cut it into 2 or 3 pieces, the middle part, internally, you call that kind of enzymes as restriction endonuclease. That means within the cut is made. So restriction enzymes are molecular scissors which cut DNA or RNA or any molecules with inside them at specific sites. Because they cut at specific sites, they are called as restriction enzymes. Okay, and they are of 2 types, which are restriction endonuclease and restriction exonuclease. Restriction endonuclease and restriction exonuclease. Okay, so they cut within and the other one cut at the end. Now let me tell you how this name restriction enzyme was coined. So this restriction enzyme, how this name was given as restriction enzyme, it's a very interesting experiment. Actually, it is a serendipity. I think you know what is serendipity. Serendipity means chance discovery in science. Discovery of antibiotics, penicillin, was a serendipity for Alexander Fleming. Same way this also, a finding of restriction enzymes was a serendipity in biology. So here, see children, I'm just drawing a vague structure of a bacterial cell, and this bacterial cell is attacked by a bacteriophage. Okay, this is a bacteriophage. Bacteriophage means they are viruses which attack a bacterium. Bacteriophage means bacteria eating. Bacteriophage, phages means eating. Actually, it is affecting the bacterium. Who is affecting? The bacteriophage. So they are actually viruses. So viruses which affect bacterium, they are called as bacteriophages. So this is how it usually affects the bacteriophage. This is the bacteriophage or the virus. Okay, and here I have just drawn a structure of bacterium in fully literal manner. I have drawn here. Okay. Now this is its genetic material or DNA of the virus. What it does is it injects the DNA here inside the bacterial cell, and this DNA keeps on multiplying using the machinery of the bacterium, and then it starts preparing the protein coat over it. Okay, So that means many phage particles are now inside. Many phage particles are now inside, and ultimately the bacterial cell will burst open and these phage particles will come out. This is the usual cycle of a bacteriophage with a bacterium. Now what scientists have observed is in certain bacterium, when a particular bacteriophage is coming and it is inserting its DNA, this process is not happening. Instead, this DNA which has entered is cut into pieces inside. It is cut into several pieces inside and the bacteria is unaffected. Then the scientists became interested in this particular thing. They started isolating these pieces of DNA. And every time they found that when this particular bacteriophage is affecting the bacterium, there is a particular, it is not always this is happening. This is happening only with respect to particular bacteriophage and bacterium. So every time they are encountering each other, these pieces which are produced will be of equal number, and the area where it is cut is every time in the same area. The same area is cut, same number of pieces, same size of pieces of DNA is generated. So they understood there is something scientific that is happening inside the bacterium, and this bacterium is having something inside it. We don't know what is that. This is a great question mark. This bacteria is having something inside it which is restricting the attack of the phage. Okay, here the phage is very well attacking the bacterium, but here something is there which is coming and cutting its DNA into pieces, the phage DNA into pieces. So the phage cannot actually attack the bacterium and the bacterium is safe, and the bacterium is restricting. The bacterium is restricting the attack of the bacteriophage. And then they wanted to find out what is this something, and eventually it was found that there are certain enzymes which are doing this function, which are doing the function of restricting the attack of phage to this bacterium. And so they termed these enzymes as restriction enzymes. Restriction enzymes, because they restricted the entry. That's how it was termed as restriction enzyme. But truly its name was very well clear for its function also, because it is cutting, it is doing the process at restricted sites. So restriction enzymes are those enzymes which cut DNA molecules at specific sites. If they cut from the end, you call it as restriction exonuclease, and if it is within, you call it as restriction endonuclease. So I told you these enzymes are present in the bacterial cell which is doing it. Now once they isolated the restriction enzyme, okay, fine. Later on they found out that inside the bacteria, inside one bacterial cell, there is not just one restriction enzyme, there can be several restriction enzymes. And scientists in different fields, they started isolating restriction enzymes plenty in number. Then there occurred a task, how to name these restriction enzymes. We cannot just name it as restriction enzyme 1, 2, 3, 4, there is no scientific basis. So there was a genuine reason, there was genuine need to find out a way how to name the restriction enzymes. Let's focus on that now. Naming of restriction enzymes. I'll give you one example of naming of restriction enzymes. We know restriction enzymes are isolated from some bacterial cell. Let me take the example of a bacterial cell, Escherichia coli. It's in simple terms, you call it as E. coli bacterium. Escherichia coli. So here this Escherichia is its genus name. Coli is the species name, or you call it as specific epithet or species name. And you know, bacterium, microorganisms, they are also specific for the strain. Here the strain is RY13. This is the strain of the bacterium. Okay, so this is the whole name of the bacterium, Escherichia coli, RY13. This is the bacterium from which, imagine I have isolated a restriction enzyme. Now I am going to name the restriction enzyme. How to name? First, I'll take the first letter of genus name. First letter of genus name. Now which is that letter? This is the genus name. First letter is E. So I have put the first name E. Okay, first letter I got. Next I'll take the first two letters of genus name. Sorry, not genus name, species name. The first two letters of species name. First two letters of species name. Now which is the species name? E. coli, and its first two letters are C and O. So I am writing here C and O. Okay. Next I'll take the first letter of the strain. First letter of the strain. My strain is RY13 and its first letter is R. So I am taking that EcoR. EcoR. This is my restriction, the name of my restriction enzyme. So I have isolated restriction enzyme from Escherichia coli RY13, and according to these rules, I have named it as EcoR. Now, from the same bacterium, E. coli RY13, I have isolated another restriction enzyme, which is making cuts at a different site. So I have isolated one restriction enzyme, I named it as EcoR. Now I am isolating from the same bacterium another restriction enzyme. Now I cannot put the same thing here, EcoR. So what do? I add Roman numbers. Now the first one that I have isolated, I'll put here EcoR1. The second enzyme I have taken from the same Escherichia coli RY13, but I have isolated it for the second time, so I'll put here EcoR2. Now what is this R1, R2, R3? What is this 1, 2, 3? It is the order of discovery. Now after that I'll put the Roman numbers. According to the order of discovery. So EcoR1, EcoR2, these are the restriction enzymes isolated from Escherichia coli RY13, and according to this rule I have named it. It's very simple. First letter of the genus name, first two letters of the species name, first letter of the strain, and then if more than one enzyme you have isolated from the same organism, you can just demarcate it with Roman letters according to the Roman numbers according to the order of discovery. I hope naming of restriction enzyme is clear to you. The meaning of restriction enzyme is also clear to you. In the next session, we'll see how this restriction enzyme works. Thank you all.